Let's look at some microevolution with Hardy-Weinberg equilibrium. In this lecture, you should be able to calculate allele frequencies in a population, be able to define Hardy-Weinberg equilibrium, evolution, population, gene pool, allele frequency, and genotype frequency, be able to list the 5 assumptions of Hardy-Weinberg equilibrium, giving an example of a violation for each assumption, which will largely be the next couple of lectures, and explain how violating each assumption changes allele frequencies, again, largely the next couple of lectures. Be able to write the Hardy-Weinberg equations and be able to define each of the terms therein. So, here are some review questions we'll cover in class and take a little while. We're going to do a little bit of review about alleles and segregation. I highly recommend that you also review these. And we're going to start with microevolution. This is the change in allele frequencies in a population over multiple generations, where an organism will be different than its ancestors because it has different alleles. And we are dealing with 3 things in this statement: allele frequencies, population, and generations. So let's look at what a population is. A population is a group of organisms of the same species living at the same time in the same place and thus capable of interbreeding. We use this as an ecological unit of study. When we get a group of organisms together, they are a population and we can study that specific population. Doing so is called population ecology or demographics. And we're going to look at something called microevolution that looks at the allele frequencies and how they change in that population. So, in order to look at gene frequencies, we have to define a gene pool. I want you to imagine a bunch of frogs. Now, this bunch of frogs lives in a forest, and in that forest there is one pond, and that pond is where they all go to spawn. What do frogs do when they spawn? Well, they put all their eggs in the pond, and they put all of their sperm in the pond. So essentially, all the gametes of that population of frogs are going to be in the same pond at the same time. This is quite literally a gene pool, where you have all of the different alleles are available in the same area. Now, with frogs, it's pretty easy to imagine, but if you imagine saying a population of pigs, we're saying that they can all just kind of share alleles as they are randomly mating. And that's the gene pool. It's all the individual genomes in a population. We can measure it 3 ways. First off are all the alleles, which are the allele frequencies. And that's what we're going to be looking at today. Next are all the loci, which gives a genotype frequency. Another thing we're going to kind of look into today. And we can look at it in terms of all individuals, or the size of the population. And that's probably going to be another class. So how do we define these allele frequencies? Well, let's look at this. The actual allele frequency is the number of copies of a specific allele in a population divided by the total number of all alleles for that gene in the population. And that is the allele frequency. The genotype frequency is the number of individuals with a particular genotype, be it homozygous dominant, heterozygous, or homozygous recessive, in a population divided by the total number of individuals in the population. Why do I say actual for both of these? Well, we're going to be looking at predicted allele frequencies and predicted genotype frequencies as well today. A population, if it has 40% tall alleles, remember that tall plant, and 60% dwarf alleles, or that dwarf plant, regardless of the genotype or phenotype, these are the allele frequencies. The allele frequencies are 40% and 60%, or 4/10 and 6/10. The population, let's say it's 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Those would be the genotype frequencies. In the second example, 25% of the population will actually show the phenotype of homozygous recessive or dwarf plant. So we're going to be looking at that phenotype to know what the genotype for the homozygous recessive is. And 2 populations can have the same allele frequency, but different genotype frequencies. And we can see that when the genetic structure is different, and that's the allele frequency and the genotype frequency when considered together. So we're going to define allele frequencies as P's and Q's. P is the frequency of the dominant allele, Q is the frequency of the recessive allele, and 1 is the total frequency of all alleles. In this case, there are only 2, so both alleles. And we know that P plus Q equals 1 because in this case, there are only 2 alleles, so the frequency of both of them must add up to 1. Back to the Punnett square. Okay, We're going to look at this in terms of P's and Q's in a total population. Now imagine all of the alleles in the population can be shared randomly. So they're randomly mixed. So we can get homozygous dominant, or 2 P's. We can get homozygous recessive, or 2 Q's. And we can get heterozygous, or P and Q. Now if we put frequencies of those alleles, you have 7/10 and 3/10 here, we can predict the frequencies of the genotypes based on the allele frequencies. So it's just those multiplied by one another. And this requires a little bit of math, and we're going to be doing that in lab as well as in some examples. And when we do this, we can use something called the Hardy-Weinberg equation, which is P squared, or those double P's, plus 2PQ, which is both heterozygotes, plus Q squared, which is those double Q's, equals 1. Why? There are only 3 available ways that you can have a genotype, and therefore they all together will add up to 1. So there's your genotype frequencies if you were dealing with something like large A, little a. And you can use this Hardy-Weinberg equation to estimate allele and genotype frequencies in a population, but only if that population is at what we call Hardy-Weinberg equilibrium. We'll discuss that a little bit more later. Right now we're going to look at some examples of math. So in class we're going to be doing the examples here. We're just going to pause them. So we're going to do the actual genotype frequencies. Now remember, the genotype frequency is the frequency of the genotype divided by the total number. Now remember, the genotype frequency is the total number of individuals with that genotype divided by the total number in the population. So for homozygous dominant, it's going to be 90 over 200, or 0.45. For heterozygotes, it's going to be 40 over 200, or 0.2. And for homozygous recessive, it's going to be 70 over 200, or 0.35. Double check this to make sure it adds up to 1, and yes, it does. So what about the allele frequencies? Well, remember, each individual has 2 alleles. So the total number of alleles is 2 times n, where n is the total individuals in the population. So 2 times 200 is 400. And the frequency of your dominant allele is going to be 2 times 90, because each of those 90 have 2 copies of the dominant allele, plus 40, because each of the heterozygotes has 1 copy, divided by 400 total alleles in that population, or 0.55. The allele frequency of the recessive one is going to be Q, and that's going to be equal to 2 times 70, because each homozygous recessive individual has 2 copies of the recessive allele, plus 40, because each heterozygote has 1 copy of the recessive allele, divided by 400 total alleles in the population, and that's going to be 0.45. We're going to check our math here. P plus Q has to equal 1. 0.55 plus 0.45 equals 1. Now, do it on your own. Pause the video and unpause it to see that, yes, you were definitely right. Those numbers do add up to 1, and that's the numbers. Now, what about the allele frequency? Pause the video and unpause it to see that, yes, you were right. Those do add up to 1. And what you're noticing is that these have different frequencies of genotypes, but they have the same allele frequencies. Remember, 2 populations can have the same allele frequency, but different genotype frequencies, and we call that genetic structure. So, we said if a population is in equilibrium, we can predict the genotype frequencies based on the allele frequencies. We have 5 conditions or assumptions that we have to ensure that a population is in equilibrium. First off is that the population is large, and small random events are not going to cause random changes. Next, that the population is isolated, that there isn't some other group of alleles coming in and changing the allele frequency by dumping alleles in our population. Third off, that there are no net mutations, that there isn't somehow a large number of homozygous recessive that are becoming heterozygous due to mutations. And that next, that there's random mating, that for say, there's just random mixing in the population, that you don't have all the homozygotes all mating with one another and the heterozygotes not mating at all. And last, that there's equal survival and reproduction, that the heterozygote doesn't somehow die off or one of the homozygotes isn't lethal. So these are the assumptions that we're looking at with Hardy-Weinberg. And if allele and genotype frequencies stay the same from one generation to the next, and there is no natural selection, there's no non-random mating, i.e. there is random mating, there's no genetic drift, i.e. the population is large, and there's no gene flow, i.e. the population is isolated, also there's no mutations, then that means there's no microevolution, no change in the allele frequencies in a population over generations, which means no macroevolution, which means no new species are being formed. We'll get into that later. So at Hardy-Weinberg equilibrium, the allele and genotype frequencies stay the same from one generation to the next. The gene pool is static, the genetic structure stays the same. If it's not in equilibrium, we say the population is evolving. And evolution, remember, is a change in the allele frequency in a population over generations. It is also known as descent with modification, as something changes as a species grows over time.